Okay, so good evening. Welcome to today's session. Today we'll be beginning the 23rd class for this course. We only have two more classes left. So, continuing with yesterday's discussion, we will be wrapping up a little bit more. We'll be learning a little bit more about chromatin accessibility profiling methods. So, before I get into the discussion for today, which will be centered majorly around two techniques, one which is high C and the other which is Kayapet. These two techniques, we've already gotten some introduction to these in yesterday's class, so we'll be continuing with that today. So if you see here, I'll just give a quick recap of yesterday's discussion so that so that we can recapitulate and we can connect it with our discussion for today. So in yesterday's discussion, what we had brought up was just a second. What we had looked at was the 3C domain or the 3C methods of chromatin accessibility profiling. And under these, what we first started our discussion with was the architecture of the genome, the 3D nature of the genome, the entire landscape, the way in which chromatin organization is something that is so important for us to study, because till now we have majorly been talking about the genome as sort of a linear structure, much like a line, like a sentence. And in the sentence, we have imagined the words to be genes, which are just arranged in a certain order. And we have, under these assumptions, studied a lot of these things. But within the cell, considering the immense role of epigenetic modifications, considering the fact that the nucleus is compacting such immense amount of information in very small space, then that compaction also, we can assume it to be biologically meaningful. Now, under that assumption, understanding what biological meaning that particular organization might or might not have becomes very important. And under that purview itself, we actually focus our just give me a minute. I'm just getting calls. Okay, sorry about the disturbance. So we're talking about the architecture of the genome having some biological meaning. And when we say biological meaning, it means the fact that if two regions or two locations, two genomic locations are present close to each other, then that possibly has some sort of a meaning, and that helps control some sort of a pathway, helps regulate the expression of certain genes, and so on and so forth. So if you consider the entire human genome, it comprises approximately 2 meters of DNA, which is compacted into such limited and such constrained space. And this is almost considered to be analogous to packing 40 kilometers of thread into a tiny tennis ball. And this process is not random, it is sophisticated, architecturally controlled process, which is essential for the cell's function. Now, each cell must constantly negotiate a dynamic equilibrium that between this packaging and the need to access the genetic information, which is necessary for fundamental processes like gene expression, DNA replication, repair, etc. Um, the 3D organization of the genome is what makes all of the difference, which is what I was just talking about. And the organization of the genome, it exists as a functional folded landscape, and it is organized hierarchically, beginning with the confinement of individual chromosomes into these little territories or these nuclear volumes, which are known as chromosome territories. Now, within these territories, the chromatin is further segregated into large-scale active and inactive compartments. And when we talk about active and inactive compartments, it is analogous to considering how the DNA is present in both the heterochromatin and the euchromatin, much like that. There are some regions which are transcriptionally active, some which are not. And at finer resolution, these compartments are built from very small self-interacting regulatory units, which are called as TADs or topologically associated domains, which in turn is shaped by specific point-to-point chromatin loops. And this intricate architecture is not just structural or it's not static in nature. It represents a critical layer of gene regulation. And by folding in three dimensions, what it essentially does is that it brings very far away elements close to each other, which are usually regulatory in nature. And these include enhancers, silencers, etc., which come in direct physical contact with their target gene promoters, which is equivalent to controlling gene expression. This is understood. The role or the importance of this organization is especially understood when there's any disruption in this architecture, when it is compromised. So this has actually been linked to a number of diseases. Majorly, two types of disorders or two types of diseases are usually affiliated or associated. The first are developmental diseases. So there can be congenital defects which pop up when there is such dysregulation or irregular behavior, versus when there is cancer, that during cancer or at the onset and the progression of cancer is also many a times driven by this. Chromosomal rearrangements, which are usually a hallmark of cancers in general, they do more than just simply alter the linear sequence. Sometimes they can catastrophically rewire the 3D landscape. For example, the translocation of a potent enhancer near a proto-oncogene, or the breakdown of a TAT boundary that normally insulates an oncogene from the activating elements, can sometimes lead to aberrant gene expression and can drive tumorigenesis. Mm, So what do we mean by that? We mean that these boundaries, the TAT boundaries that I was talking about, they can insulate some activating elements. So they can, if you remember, we had talked about when we were initially discussing gene regulation, we had talked about the fact that there are certain regions of the genome which sort of insulate signals or which can quench signals. And they define these boundaries, some regions which are active and some regions which are not. And these activating elements are usually protected or kept away from oncogenes, so that tumorigenesis can be to some extent kept at bay. And for much of the history of molecular biology, the 3D genome has remained largely invisible, accessible only through low resolution lens of microscopy, which lack any sequence specific detail. This has changed quite dramatically with the development of the 3C method and its derivatives, which have collectively been the driving force, like we've already discussed. So what does it exactly do? It converts the physical proximity of some genomic loci, usually which is transient or dynamic in nature, into something which can be considered to be astable, uh, be quantifiable, and three, the product of ligation between two regions. So this conceptual leap actually bridged the gap between physical structure and genetic sequence, which allows researchers to, for the first time, create very high resolution maps of the folded genome. So this is a representation of chromatin or chromosome capture assays, which an example of which is 3C qPCR. So if you look here, this is your chromatin loop, which has two regions brought close together by the interaction of certain proteins. Then cross-linking is allowed to proceed, which will freeze these interactions, DNA-protein interactions in place. So these are your candidate interacting fragments. You have this pink region and this blue region and this red and this green, and among them and farthest is the black. And as a result of looping, there are certain regions which are able to interact with each other. Largely, that includes sorry, the black and the red regions. So after 3C assay, what do you see? You see that this linking or these interacting regions are still in contact with each other, while the top of the loop is chewed up or is digested. After digestion, what happens? This using a small linker, these two regions can be then ligated, and then qPCR can be performed to quantify how much of these two interacting regions can actually be found there. So this 3C protocol, like we already discussed, the idea was that you simply want to understand how do two regions which are quite far apart physically able to interact within the 3D space of the nucleus. The protocol's genius lies in the method of converting that physical property by physical property of spatial proximity into a genetic one, which can be detected and quantified as well. And it is a multi-step process, a biochemical process also at that. And through variations in the workflow, the core workflow remains mostly similar, and we'll discuss the steps one by one. It starts off with in vivo cross-linking. So usually, the common cross-linker agent or cross-linking agent which is utilized is formaldehyde, and it is directly added to this cell culture medium. What does it do? Formaldehyde permeates into the cell and the nuclear membrane as well, creating these covalent DNA protein and protein cross links and freezes those interactions in place, which is similar to sort of taking just a quick screenshot or a snapshot of the entire region in 3D confirmation. And also, what else it does is that it limits the dynamic nature of these interactions so that it can be successfully captured at that point of time. So, yeah, now this step needs a lot of standardization. Yeah. As over cross-linking can sometimes create very large insoluble DNA protein aggregates which are resistant to enzyme digestion. Therefore, it reduces the efficiency of the entire protocol. So post-standardization, it is going to be a piece of cake. Next comes chromatin fragmentation. So after cross-linking has been done, the cells are lysed, nuclei is isolated, and the cross-linked, yeah. Um, for chromatin fragmentation, following cross-linking, cells are lysed and nuclei is isolated. Cross-linked chromatin can then be digested with a restriction enzyme like Hindi3 or EcoR1 as well. And after digestion, what happens is that you get these two open ends, which can then be ligated as well. And because these two regions are spatially proximal under those circumstances, under whatever conditions you're performing the experiment, they remain physically tethered together by whatever ternary protein complex has facilitated that interaction. Oh, sorry. Then comes linking the interaction among chromatin. So this is the conceptual and the technical core of this method. So the mixture of cross-linked digested chromatin fragments is subject to ligation with DNA ligase under extremely diluted conditions. The high volume of this reaction makes it sort of ensures that the concentration of chromatin complexes is very low. So the probability that two fragments from two different complexes are randomly colliding and getting ligated together is, of course, almost an improbable event. And instead, any such transient proximity must be regulated by the cell and or can be predicted to be regulated by the cell and predicted to be a necessary event. So the reaction, preferably because you have diluted the reagents to such an extent that non-specific ligation will not take place. As a result, the reaction favors ligation between those fragments which are already in close proximity with each other, which is actually how you kind of put a checkpoint to ensure that whatever results you're getting, they're not just, simply not just fallacies. Analysis and quantification. So after ligation, the crosslinks are reversed, typically by heat and by treatment with proteinase K, which degrades proteins and releases the DNA. And what you get as the resulting library, it consists of a mixture of religated fragments and the chimeric ligation products of interest. So the original 3C protocol uses the qPCR method with a pair of primers, which specifically amplify the junction between these predetermined genomic loci. Then how much PCR product is generated can be quantified, and that should be directly proportional with those loci that had interacted in the original cell population. So this is a sort of a schematic representation of pretty much the same. I'm not going over it. What I would like to bring to your attention is the fact that these techniques, all these often parallelly mentioned techniques, they have similar outputs, but not the same. So if you look at the techniques one by one, the first is 3C. In 3C, you get a one-on-one interaction. That is region 1 interacts with region 2 specifically. That information you can get by validating it using qPCR. If you opt for 3C-seq, which is the sequencing associated protocol, in that case, you can get a more comprehensive information from the same experiment. Second is 4C, where we have one versus all. So you have one locus and you find all of these putative interactions with all these different loci, and that gives you a one versus all comparison. When we talk about 5C, that is a many versus many. So you get a more comprehensive, more high throughput result from it. When you talk about QIAPET, then it is involves a protein as well. We'll discuss about it today in a little more detail. And its strategy is also a little bit different in this case. And then you have high C, which gives you an all versus all comparison. So we'll discuss that also today. So, 3C uses PCR primers, like I mentioned. 4C employs a second digestion step followed by circularization, which is why sometimes it's also considered to be circular 3C qPCR, which is followed by inverse PCR from a known bait fragment so as to identify all its interaction partners. Then 5C uses a multiplexed ligation mediated amplification with universal primers to detect all interactions within a large defined region. Then it incorporates biotin labeled nucleotides during the filling in step, which allows for the enrichment of ligation junctions and subsequent genome wide sequencing. So, yeah, you see, again, this shows you more detail about the protocols like we've just discussed. But if you see, the initial few steps are the same for all of these 3C techniques. So you have cross-linking of chromatin, you have digestion of the cross-linked chromatin, you have ligation of these proximal regions, and then reverse cross-linking, and you can infer a lot of different kinds of information from this data set. Next was QIAPET. We'll discuss about it today. So, okay, let me just go over it quickly. So in QIAPET, this technique, which is chromatin interaction analysis by paired end tag sequencing, which is QIAPET, what does it involve? Of course, if there's involvement of proteins, like I just mentioned, then that necessarily includes an immunoprecipitation step, which will allow us to map any long-range interactions, much similar to that of high C. So in this method, what happens is that the DNA protein complexes are cross-linked. They're cross-linked and they're fragmented. Then some specific antibodies are used to immunoprecipitate the proteins of interest. And two sets of linkers are utilized, which have unique barcodes. Again, we'll learn more about it in detail today, so I'm not going over it. I have much better schemas, which I'll use to explain it. And they have unique barcodes and they're ligated to the ends of DNA fragments in separate aliquots. And then they're able to self-ligate based on how proximal they all are. Now, the DNA aliquots can be precipitated, digested with restriction enzymes, and sequenced. The deep sequencing provides base pair resolution to the ligated fragments. HiC and QIAPET currently are providing the best balance of resolution and reasonable coverage in the human genome so as to map these long range interactions. So, what are the pros? What are the cons? Pros, very suitable for detecting a large number of long range and short range chromatin interactions globally, studies the interactions made by specific proteins or protein complexes as well. Um, Public QIAPET datasets are available through the ENCODE project. You can use the ENCODE project to access it. Then removes the background, which is generated during traditional chip assays. And the immunoprecipitation step obviously removes a lot of non-specific background and reduces data complexity by a lot. What are the cons? It requires a large amount of starting material, generally, one second. Generally, 100 million cells are required. Non-specific antibodies, if present, they can pull down unwanted protein complexes and contaminate the pool as well. Yeah. Um, Linkers are capable of self-ligation, much like primers. They can generate an ambiguity among true DNA interactions. Limited, very limited sensitivity, or less than 10% interactions might get detected. So that's what you see here. You have sample fragmentation followed by immunoprecipitation, ligation, and then restriction enzyme digestion. We'll see a little more of the videos today. Then we moved on to 4C and 5C. I don't want to repeat it again. Then the applications of these techniques were also discussed that it usually gives you a high resolution 1D profile of the interaction profile of the entire um, genome of the interactomes, which involves certain genetic loci. And this makes it a very invaluable tool for discovering novel long-range enhancers which regulate your gene of interest, for identifying target genes of some non-coding diseases associated SNPs, or to characterize how the nuclear environment of some specific loci change during cellular differentiation or in response to certain stimuli. For example, 4CC has been instrumental in mapping the complex interaction landscapes of oncogene enhancers, revealing how they can, sort of supersede or hijack regulatory control over some distant genes inside cancer cells. Um, Following this, we move on to our discussion for today. We'll be looking majorly at these two techniques, the high-seq method and the chimet method. So let me start off with this image. Okay, so most of the information that you will find in this slide and the next are sort of composite overview of everything we've discussed. Not just what we've discussed, but it goes beyond that as well because it encompasses strategies that the concept behind these strategies and why we are so bothered about it. What is the biological relevance of it, and why are they so prevalent in today's time? What is the reason for the extremely high cost that is associated with these techniques, and so on and so forth. So in ATAC-seq, the hyperactive version of the TN5 transposase is used to preferentially insert into accessible chromatin while simultaneously attaching adapters to resulting fragments that can be used to directly amplify sequencing libraries. So let me just repeat it once again, even though we've already discussed ATAC-seq. But what is the main difference? The main difference is the presence of a completely different enzyme, which is a hyperactive TNK transposase, which stands for the T in ATAC-seq. And what it can do is preferentially it can insert um, into into regions of the chromatin which are open and attaching adapters to those fragments, which is then used for library sequence amplification. In the DNase-seq technique, which is similar but not exactly the same, um, in DNase-seq, the DNase-1 enzyme is used to preferentially cleave this accessible chromatin and generate fragments, which you can then, amplify into sequencing libraries. Now, both ATAC-seq and DNA-seq are generating peaks in read coverage over accessible regions in the genome. Then you have MNase-seq, which is micrococcal nuclease sequencing method, which involves the use of the MNase enzyme to digest unprotected DNA, leaving behind intact fragments which are protected by protein occupancy, majorly by nucleosomes, which are then amplified, which are then amplified, resulting in increased read coverage over positioned nucleosome. And in MNase-seq, what do you do? The enzyme is completely different. In DNA-seq, of course, it's DNA is one enzyme for digestion. In ATAC-seq, it is the hyperactive TN5 transposase. And in MNase-seq, unprotected DNA is digested, leaving behind intact DNA fragments, which have nucleosome protection. Um, the methyltransferase based approaches, which include NomiSeq, DSMF, SMAC-seq, NanoNomi, MESLMR-seq, and Samosa. These are all different assays which involve certain strategies. Just give me a minute, please. Yeah. So, I was saying that these modifications, which can include M5C or the methyl 5-methyl cytosine methylation in CpG or GPC islands, um, these modifications can be present there as well, and also M6A methylation. Now, bisulfite conversion or the EMC assay converts fragmented DNA. Again, don't bother about it too much. A lot of it might sound a little too heavy, a little too informative, but it's okay. Yeah. Alternatively, long read sequencing, which can also directly read. Alternatively, long read sequencing, which can also directly read M6A methylation and take advantage of its much higher density in the genome, can be used, resulting in multi-kilobase scale single molecule footprints. Methyltransferase-based approaches tend to provide a simultaneous readout of both nucleosomal positioning and open chromatin regions, appearing as small bumps in the methylated fraction of bases over linker regions and larger peaks over regulatory elements specifically. Okay, so coming back here, we have not at all introduced these methods, so I'm not going to go into details about it, about methylation associated strategies, but our interest lies largely in chromatin accessibility and nucleosomal positioning. So that is pretty much what we'll keep our attention on, but I just wanted to summarize the rationale and the logic behind it for you to understand. So we will begin our discussion on the high C method. So the high C method or technology, it stands out as a powerful, extremely relevant and informative technique to investigate the 3D structure of genomes. Um, it is a sort of a hybrid or the fusion of two strategies. One is high throughput sequencing, obviously. The other is 3C, which is chromosome conformation capture, and integrating both of these, HiC offers insights into the spatial organization of chromatin within the nucleus. So 3C involves a couple of steps. First is fixing the nuclear chromatin, digesting chromatin protein crosslinks, ligation of digests, the release of bound proteins, and PCR analysis to detect interactions between DNA fragments. Assumption in this method is the fact that physically those DNA fragments that are close to each other will exhibit much higher linkage frequencies, which can be studied using locus specific PCR. So what are the advantages of this high C chromosome construction? So um, it individual centric approach. So unlike traditional methods that often require constructing populations, high C chromosome construction can be done or completely executed by a single individual, and this streamlines the process, reducing the need for extensive population based studies. Enhanced localization efficiency, high C chromosome construction. It boosts higher localization efficiency compared to many conventional techniques, which means that it can specifically give you the spatial organization of genomic sequences, and it can give you detailed insights into like the chromatin interactions, the structural arrangements, and all that. Um, Error correction is much better. There are capabilities. Yeah. The capacity, one major notable advantage of high C chromosome construction is the capacity for error correction during the assembly of the genome, which allows researchers to refine and improve the accuracy of the assembled genome and ensuring reliable results for downstream analyses and interpretations. So if you look here, you'll see that these are different types of chromosomal errors that can potentially exist. You have chromoplexy, you have chromothripsis. So like you can see, there is chromosome A and chromosome B, and there are multiple breaks in the chromosomes, both shown in blue and green respectively. And as a result of NHEJ and alternative end joining, what has happened is that these gaps have been filled and sort of rectified, but they have now included a portion or a fragment of some other broken chromosomes, and that can be extremely dangerous for the cell. That can be cancer inducing, it can be self lethal, it can be a number of things, but that is what happens under the purview of chromoplexy. The other one, which is chromothripsis, it occurs under extreme catastrophic chromosomal breakage. Um, so as a result of which, as you can see, the arrangement of the entire chromosome is completely um, disturbed is also a subtle word, but is imperfect, inaccurate, and random, in this case, random chromosomal repair by NHEJ. So some portions of the chromosome are actually lost, some of it is retained. Hmm. B is for fibroblasts and LCLs. have Illumina GS, PacBio GS, IC, RNA-seq, etc. You do structural variant calling, genome reconstruction, haplotyping, allele-specific gene expression analysis, 3D chromatin structure of reshuffled alleles, etc. Here you see these chromosomes between chromosome 2 and 16. There has been some exchange, so that can be detected as well. And here you have chromosome 2 on the x-axis, and you have chromosome 2 also on the y-axis, and you can check this is the result of IC data, which looks somewhat like this. Let me show you the video which has been shown here for better understanding, and then we can come back to the text um, for more clarity pretty soon. Yeah. If you see here, it looks like there are two subunits which are holding these two regions of the genome close together. That potentially can be some part of the entire complex which facilitates this interaction. And you lyse your cells and you get your DNA. You subsequently fragment it with some sort of restriction sign like KD3 or require1. Now, keep in mind that this step may or may not be present there. It depends upon what sort of ligation or what sort of library you are using. In this case, the library they've suggested involves the use of biotinylated primers, which are able to bind to the sticky end or the overhangs. And then after this binding or after this biotinylation step, they can then be ligated to bring them close together and so that is the logic. So that is the logic behind using extremely diluted amounts or diluted concentrations of our target enzymes, and it allows blunted ligation to proceed. Then the library can be shared. The junctions that you get can be then pulled out using streptavidin beads, which bind to avidin, or sorry, to biotin, and then they can be pulled down by that way. So that was pretty much. Um, yeah. So if you look at the workflow of IC sequencing technology, then first step is that cells need to be cross-linked. So cells undergo preparation and fixation through cross-linking with either paraformaldehyde or formaldehyde, which preserves intercellular DNA protein and DNA interactions, maintaining the 3D structure inside the cell. So, for example, for live samples, a typical treatment involves 1 to 3% formaldehyde for 10 to 30 minutes. Yeah, at room temperature. However, it's crucial to note that this can potentially hinder the efficacy of DNA sequence digestion by restriction endonucleases, and it requires very precise control. So first step, of course, is cell cross-linking. You have to cross-link with paraformaldehyde. You have to quench it using glycine, preserve these intracellular DNA protein and DNA interactions, maintaining the 3D structure. Then 1 to 3% formaldehyde can be used for 10 to 30 minutes. And second step is endonuclease digestion. So typically DNA gets enzymatically cleaved using restriction endonucleases, generating sticky ends on both sides. Like I just mentioned, EQR1 or Hindi3 enzymes like this can be used, which will enzymatically clean up or will enzymatically cleave using restriction enzymes. And the sticky ends usually can be ligated, can often be biotinylated, especially at the overhangs. And the size of the resulting fragments end up influencing or impacting sequencing resolution also. Generally, there are two enzymes that are used for this process. So, or for this selection, there's a six base pair restriction endonuclease, or it's just, yeah, let me take the Yeah, so generally there are two enzymes that are selected. One, a six base pair recognition motif containing enzyme, or a four base pair considering restriction enzymes. Four base pair restriction enzymes will work differently, and as a result, you do not expect to see any overlap. Enzymes like EQR1 and Hindi3 are used to cut the genome approximately at 4052 base pairs or approximately 4000 base pairs, resulting in approximately 1 million fragments within the human genome. So this is your cross-linked chromatin, which has been cross-linked with something like paraformaldehyde, for example, following which restriction digestion is done. Using these two enzymes that we just talked about, a six base pair and a four base pair. Six and four are the length of their recognition motifs. Then biotinylation is carried out. Proximity ligation is then carried out, where based on the proximity between these two places, ligation can be performed and then flow through. Then, like we just mentioned, these fragmented DNA sequences, they will undergo end repair to get a continuous sequence, which can then be used for downstream analysis. So either flat ends or sticky ends can be generated, which undergo repair to create these blunt ends, during which process biotin-labeled bases can be introduced to facilitate subsequent DNA purification and capture as well. Cyclization and repaired DNA fragments are looped between DNA segments which contain interactions using T4 DNA ligase. So subsequently, proteins which connect the DNA fragments can be digested to isolate the cross-linked fragments. So what is happening here? You have till sorry till the previous step, you had cells which you lysed, you fixed, preserved the DNA protein interaction steps. Then you subject that DNA, the now fixed DNA, to endonuclease digestion or restriction enzyme digestion, which generates sticky ends at both sides. So it keeps on chewing up and it generates sticky ends. These two sticky ends, uh, they undergo proximity ligation, and the two type of enzymes which are selected usually are such that one is six base pair recognition motive specific enzyme, the other one is a four base pair, and so on and so forth. Next, in the subsequent step, end repair is carried out, whereby the fragment in DNA, it possesses either sticky ends which undergo repair, they make blunt ends, and during this process, biotic labeled pieces are introduced to facilitate subsequent DNA purification and capture. Um, Next, during cyclization, end repair DNA fragments get looped between DNA segments which con- which contain interactions using P4 DNA ligase, and subsequently proteins which connect the DNA fragments can be digested. So you have your end repaired fragments which can be looped between DNA segments which contain interactions using P4 ligase, and then they can be digested to isolate the cross-linked fragments. The DNA is decross-linked, purified, and fragmented into 300 to 700 base pair fragments. Those that contain interactions are then captured for library construction using strand affinity magnetic beads. So, ultrasound or similar methods can be employed to further break down those fragments. And finally, pattern containing fragments can be captured on magnetic beads, libraries can be constructed, and sequencing can be carried out. So when we talk about high C method, what do you get as your output? So your biotin containing fragments have now been sequenced, and now the sequencing data needs to be analyzed. So what steps does it undergo? First, of course, you do a quality control check. You filter out those low quality reads. You filter out uh, any errors, any anomalies, similar to standard second generation sequencing data processing procedures. Second, you do a sequence comparison utilizing the paired end mode, which is recommended for high C data analysis, facilitating accurate comparison of your sequences. Third, you can position the cleavage sites. So after filtering and after comparing your sequence, you position your cleavage sites. So after determining where the read pairs are present in the genome, next step is to identify the nearest restriction enzyme cut site corresponding to this read pair, which is achieved by considering the insertion fragment size restriction. The position of these cleaved segments provides the location where DNA interactions had originally occurred. Then screening for valid comparison fragments. Valid comparison fragments are identified as paired reads located at opposite sides of the opposite ends of the cleavage side and mapped in opposite direction, ensuring that there's an accurate representation of both the chromosomal interactions. Now, somebody might ask, why are we doing this in opposite directions? The reason behind that is that we expect that if the fusion has occurred between these two between site A and site B, then from the paired ends, um, like then you just want to confirm that chromosomal interactions have brought which regions from these two sides close together, and you want to confirm that both their orientations are still intact. Integrating the DNA fragment interaction intensities, the intensity of these interactions can then be sort of integrated, providing insight into the strength or the frequency of interactions between these genomic loci. And finally, you normalize the DNA fragment interaction matrix. Um, Normalization is performed to ensure unbiased comparisons and accurate interpretation of the interaction data, which is done to ensure that there are unbiased comparisons made and the interaction data is properly interpreted. So the high C assembly is typically conducted using software like LHSs, which segments, sequences, and orients the genome based on the support which is provided by these read pairs. So that includes manually mapping and verifying the genome to obtain a final chromosome level assembly. So if you look here, just another schematic to explain the same similar thing, but the difference being that this is high C for at a single cell level, so that simplifies things for you further. So you have your consider this to be one chromatin loop that has been taken forward, which has undergone cross-linking at this step. So permeabilization is done, restriction digestion is done, following which at the overhang biotin labeling is done, which is also sometimes called as the fill-in step. Then ligation is around to proceed to repair those broken ends, and single cell isolation is then performed and cross-link reversal is carried out. Then, after binding it to the beads, what sort of beads? These are streptavidin coated beads because we had a biotin conjugation. After a second digestion, A-tailing and adapter ligation is carried out on the beads. Library amplification is allowed to proceed. Size selection, quality control, quantification of the library, all of that is then carried out. So, so IC assembly is done using this LHSS software, which gives us manual mapping and verification. Then valid read pairs, they yield signals on the map with the strength of the signal being directly proportional to the spatial and the sequential distance between these contexts, which allows to correct errors, which including the identification and connection of misassembled contexts, adjustment of contact orientations, and determination of contact placement within chromosomes through clustering. Ultimately, what you get is a refined chromosome level genome assembly with any remaining discrepancies, the fine-tuned manually, with any remaining discrepancies fine-tuned manually to ensure accuracy. The manual adjustment process, it aims to achieve a clear diagonal signal indicative of a well-assembled genome. The Kaya PET method, um, cell culture is what you start off with, of course. You prepare the chromatin, you use the major distinction point is here you rely on your protein of interest. So your protein of interest is brought into question because you utilize a certain antibody, which is specific to some epitope on your protein of interest, which is available even when it is interacting with DNA, which means it's a non-data binding uh, domain, which is then used to enrich specifically for your protein of interest. Following this, two separate aliquots, for example, are weighed. One has linker A, the other has linker B. So linker ligation is the next key step, whereby you split the chromatin into two aliquots and you add two different linkers to both the aliquots. So, for example, here it's linker B and linker A. Um, yeah. Next, what do? You perform proximity ligation, whereby you mix the chromatin and elute them off the chip beads and conduct dilute ligation. Then you digest with MME1 restriction enzyme to create tags. MME1 is a restriction enzyme which is able to create tags. Same linker or different linker pets are both possible, which is then used for and data analysis. Just give me a minute. Let us look at this video and then I'll come back to pipette in details. Okay, so apart from the videos that I've shown, you can also go through these sort of lecture videos, which are going to teach you a lot in detail about these techniques. StatQuest has a lot of these videos. You can look at ISAVE, all by all interactions, levels of gene ontology, topologically associated domains, tags, etc. You can learn about all of this in a lot of detail, and they're wonderful resources for your personal, if not anything, then your personal studies for learning more about these techniques, for becoming more technologically advanced and staying more relevant. Let me put it that way. So, anyway, moving forward, we will look at the KaiPet or the GeoPet or the KaiPet method. So the outline of the KaiPet library construction method, what does it include? First, of course, is preparing the chromatin. As you can see, this is another representation of what we have already discussed. So cross-linking and then sonication needs to be performed, following which you will have these loops. You will have some chimeric pets, you will have some experimental pets, and then what do? You perform biotinylation also, and following that you perform sequencing. So if we go by the legend, sorry, yeah. So chromatin samples are cross-linked, sonicated, and immunoprecipitated. Separate aliquots of chip DNA are then ligated to barcoded half linkers, and then proximity ligation is carried out. Pets are released by restriction digestion, purified on streptavidin-coated magnetic beads, and ligated to adapters for NGS. Then experimental and chimeric pipets are examples which have been shown here. The AB linker composition of chimeric pets, the tags flanking the central linker sequence are read out and then mapped to the genome, which are identified by clusters of overlapping pets. Singleton pets indicate non-specific ligations that do not represent the true binding location or binding site of site or interaction. Okay, so that sums up what I wanted to discuss for today. And I think because these are quite advanced topics, I wanted to I want to take a pause right about here. We have two topics left, genomic and epigenomic biomarkers in clinical diagnostics and prognostics. And I will end with integrating multiomics, a comprehensive review as well. Both the classes will be wrapped up in the next week so that we can then be done with the syllabus for this course. However, as of today, I just want to quickly go through. It will not be as detailed as I mentioned today, but I do want to revise some of the topics that we had discussed uh, so that I can have an exam on it, hopefully, or at least just for recapitulating. So, in vitro and in vivo DNA protein interaction studying strategies, I just want to discuss these two, and I'll just begin the discussion in 2 minutes. Just give me 2 minutes. Okay, so back. We will just wrap up the discussion in another 15, 20 minutes. So let me just share my screen in full screen mode. Oh, sorry, not this. Okay, so if you remember, we had discussed that DNA protein interactions are extremely crucial events that take place in a that are not events, but they are dynamic interactions within the cell, which are of utmost importance. Why? Because they are necessary to um, regulate almost every process inside the cell. So that includes chromosome maintenance, transcriptional regulation, replication, DNA repair, etc. The activation of genes by DNA binding proteins is a very fundamental regulatory mechanism that involves chromatin modifying complexes, transcriptional complexes, etc., to initiate RNA synthesis. And when we talk about DNA protein interactions, it starts from something as little as just cutting it. If you think about it in the lab, even if you don't go and do such crucial processes which are necessary for cell survival, but even within the lab, when you want to chop up DNA, what do you need? You need a DNA protein interaction to actually occur. So your restriction enzyme should be able to recognize and bind to the region of your interest, and they should be able to cut within it and then execute the job which is expected of them. So while we don't really pay much heed to it, these processes are extremely crucial, extremely well regulated within the cell, and are very important for practical uh, work and for translatability in our for translatability. Let me just put it like that. So this is an example where the crystal structure of the FEN1 protein bound to its DNA has been shown. This shows you just a space filling diagram of the interaction. What are some of the techniques that you can actually use to study these kind of interactions? The first example we talked about was the footprinting assay. So in footprinting assay, basically the DNA if I just summarize and remind you of the process, the DNA is completely digested. Any region which is protected by the binding of some protein onto the DNA will be protected. Any region which is protein bound will be protected from this digestion, and as a result, will not undergo. It will be protected from digestion, and as a result, will leave behind a footprint. So, if you look here, is your isotope labeled site. And as you can see, compared to your control condition where you get fragments of all possible sizes because there was no protection in that place, the other lane or the other axis is where you see a region has remained protected because there has not been any labeling in that location. And then you can actually trace back which when I say not no labeling, it means that this region has not undergone digestion because enzymes were not able to access it, and that region sort of got protected. And the protein protection zone can then be identified to recognize the footprint. So, a sequencing ladder, footprinting reaction, control reaction, all of this is just going to give you the same idea. Also, one more thing to keep in mind whenever you do an experiment like this, you need to have a plus protein and minus protein condition, which means your control versus your treated condition. And in both cases, you should also make sure that your DNase 1 is present at extremely diluted concentrations also, such that one cut per DNA molecule is made. Obviously, you cannot ensure it, but this is how we talk about the stoichiometry, that if you have 10 molecules of DNA, then how much enzyme do you need to actually to make sure that only one cut is made per molecule? It does not always need to be a 1:1 thing, but it stoichiometrically has to make sense based on the activity of that particular enzyme. Then these are DNase 1 hypersensitive sites which are accessible to DNase 1 and it can chop them up. This is also the technique that I talked about later on, apart from ATAC-seq, another method. So footprinting has been a very valuable tool for elucidating sequence specificity, the dissociation constant of a number of different ligands which bind to DNA. The agent that is used to cleave DNA is called the probe. So the tinier the probe, the higher is the resolution provided. But the chance of cleaving DNA under the bound protein also keeps on increasing. The enzymatic digestion method includes use of DNase 1, MNase, uh, methidium, propyl, EDTA, iron, copper, phenanthroline, uranyl photocleavage, hydroxyl radicals, and iron complexes. Comparison between different probes used provide useful information on their relative merits and demerits. DNase 1 footprinting is most commonly used for this assay. It's a double strand specific endonuclease, which means it binds to the DNA minor groove, cuts the phosphodiester bond. It was developed by Gallus and Smith for visualizing the lac repressor binding to its operator sequence. It employs the use of a single end labeled DNA fragment, which is then incubated with a purified protein under appropriate binding conditions, allowing the protein to bind to its specific sequence. And then it's subject to DNase 1 treatment in an appropriate buffer with a number of different concentrations and time periods. Both the samples are then run on a DNAT gel or a denaturing polyacrylamide gel and then imaged. Like I mentioned, that's what the output looks like. Now, what are the points that need to be kept in mind? First, when you alter the experimental condition, you make sure DNase 1 makes only 1 nick per fragment, like I already mentioned, and you do that by sufficiently or appropriately diluting its concentration. Uh, yeah, DNase 1 has partial sequence specificity, which means that there are some sites which are hypersensitive and therefore they show more intense band. Second, you have to end label the DNA, and that also serves a purpose. So, in a single reaction, in one single reaction, DNase or DNase 1 will cut both the strands, which will give you a mixture, right? You'll get positive strands and negative strands both, and then that is going to completely mess up your data and make it very inconclusive. So you should radio label or tag your DNA, fluorescently label or radio label, or just simply tag your DNA to make sure that which of the two antiparallel strands does the protein under concentration actually bind. So you'll get an idea about the way in which the protein accesses or binds to your DNA, accesses and binds to your DNA, recognizes its motif, and then does its job. Um, in a 5' labeled reaction, only this 5', 3' strand sequence information is provided on the final exposed film. Subsequently, it is quite common to digest both the positive and the negative strand labeled fragments in separate tubes and then run them alongside each other. Lastly, there are a number of different methods to analyze the final footprinting image, which range from visually inspecting, creating a differential cleavage plot on the basis of densitometric analysis, etc. DNase 1 isolated from bovine sources is actually an endonuclease that cleaves both single-stranded and double-stranded DNA and generates these 5' phosphodinucleotides. And 5' phosphate oligonucleotide. So DNase 1, it cuts dsDNA in a sequence, non-specific manner. Instead, it relies or it actually requires divalent cations for this purpose, both calcium and magnesium or manganese as cofactors for full double-strand cutting activity. Just a minute. In In the presence of Mg2 plus alone as a cofactor, DNase 1 exhibits nicking activity, and the single strand nucleolytic activity is more discriminatory. So if you see here, manganese and magnesium, both of them can serve as cofactors, but the type of fragments generated will differ in each case. In case of manganese, you will get these sort of cuts. So Mg2 plus alone, DNase 1 exhibits a nicking activity. So only a single strand of the double-stranded molecule is actually cut. And in case of manganese, it is a double-stranded cut. So it cuts both of these strands. Third, the purpose of a denaturing gel is to make sure that only single-stranded DNA is what you actually eventually see. Often it is chemically sequenced and run on the same gel. Yeah. And run on the same gel as a marker to find the exact sequence of the footprint. However, some precautions need to be considered. Firstly, it's necessary that we don't titrate too much DNA within the protein sample. So a lot of the protein will be unbound and susceptible to DNase 1 attack. This will also show up to the place of the usual footprint, which will make it quite confusing and difficult to interpret. Second, the incubation of your crude sample requires competitor DNA also. Why? So that any non-specific binding, any proteins which bind non-specifically do not end up showing up as a footprint. And the limitation of this technique is that it does not provide the protein's identity. Now, because of the large molecular weight of DNase 1, um, just Yeah. So apart from proteins, footprinting is also used to elucidate the binding of other molecules like drugs to DNA. The modifications of this technique use automation and capillary electrophoresis along with fluorescent labeling, a technique using DNase 1 digestion followed by sequencing called as DNase seq is often used for genome-wide studies. Protocols that use automated infrared sequencers allowing long-range and highly sensitive footprinting have also been developed. Also, using streptavidin-bound oligonucleotides for protein binding and subsequent DNase 1 digestion makes the technique much more convenient. This can be used to fish out a protein of interest from a fruit sample, etc. Moving on, I'm not going to spend unnecessary time on it. Next technique to study DNA protein interactions involves the use of a strategy known as EMSA, which is electrophoretic mobility shift assay. So, in this case, what do? You expect to see a shift in the electrophoretic mobility of your DNA protein complex in vitro. So, yeah. So its novelty actually lies in its application to deduce the binding parameters and relative affinity of a protein for one or more DNA stretches or to detect proteins nucleic acid interactions with the aim of comparing the affinity of different proteins for the same sites. So what does it assume? It assumes that DNA protein complexes are heavier and they move slowly compared to non-denaturing when subjected to non-denaturing polyacrylamide gel electrophoresis as compared to unbound free throw. Now, since how much DNA migration is shifted or gets retarded at the time of when it is bound to protein, that gives you a super shift. So you can basically detect that if there is DNA protein interaction, migration or electrophoretic mobility will be retarded. So your complex will move much slower and you'll be able to see a band which is much higher compared to your control sample, which does not have that protein of interest. And also, if you incubate it with antibodies, which are specifically a protein of interest, and because antibodies are also proteins which are of a certain molecular weight, therefore they will show retardation by that. They'll also face retardation to the movement because these are much larger complexes, and they'll find it more difficult to find their way through all of these uh, through the gel pores and then move through it and actually overcome retardation and move through the gel. This is the example of the experiment. I will not go over the details of it, but basically what you can see here in this region is that there is a difference in the binding and apart from just simple retardation, you can also figure out the exact sequence to which it also binds by carrying out single base mutations as well. Um, can do proximity ligation assay also, and that pretty much summed up our discussion. So I think I'll stop the discussion for here for today. In the next class, we will discuss our last two remaining topics, like I've already mentioned, and that will be conducted on probably Tuesday or Wednesday, and I'll share a mail on that. So thank you for joining. I'll end the class today. Good night.